Hello, and welcome to Spotlight on Antibodies, a podcast from BioIntron, where we bring you the latest antibody industry trends. I'm your host, Jovina Solistio, and this month in July, top news includes computational tools for nanodiscs, antibody drug conjugate development, and antibody antigen research. Nanodisc platforms have emerged as an approach to therapeutic antibody discovery and drug development, especially for membrane protein targets, such as cell surface receptors, transporters, ion channels, and membrane-bound enzymes. Recent research highlights the full potential of therapeutic antibodies directed to target complex membrane proteins. A new review by Yao and Thompson describes nanodiscs as providing a native-like lipid bilayer environment for embedding membrane proteins. They stabilize membrane proteins by wrapping around their hydrophobic regions to keep them soluble in aqueous solutions while retaining their functional and structural properties. These self-assembling structures are typically formed using membrane scaffold proteins, MSPs, derived from apolipoprotein AI along with synthetic or natural lipids to fine-tune lipid-protein interactions in the target protein. However, nanodiscs can also be stabilized by other amphipathic structures, like Salipro, a lipid-binding protein-based belt system, peptides, or synthetic polymers. Advantages include display of native epitopes for antibody generation and binding, homogeneous presentation of membrane proteins for screening and antibody selection, stability for large multi-subunit complexes, for antibody discovery against targets previously thought too complex, and ability to work with native-like proteins for the drug discovery process. Another paper highlights advanced applications of nanodisc-based platforms for antibody discovery, where nanodisc technology provides a great advantage in a synergetic combination with cell-free protein expression systems. Typically, when expressing membrane proteins in cell membrane or inclusion bodies, detergent must be used to extract or solubilize the target protein, even though detergents can disrupt the spatial structure of the protein. Varying the lipid composition of nanodiscs could provide more successful insertion and folding as better native membrane mimicking properties. As depleting agents, nanodiscs reduce selection rounds and have been instrumental in targeting specific conformational states of proteins. Advances such as scaffold biotinylation and direct targeting of MSPs with antibodies further enhance nanodisc utility in antibody discovery. A few examples of nanodisc applications are described in another recent review. For endothelin receptor A, ETA, a class A GPCR implicated in various cancers, nanodisc reconstituted ETA enabled the isolation of high-affinity scFv antibody that inhibited ETA signaling and tumor growth in vivo. Similarly, SMA nanodiscs presenting the extracellular domain of parathyroid hormone receptor 1 allowed identification of an scFv HFC antibody, aiding in the study of receptor signaling bias. For the apelin receptor associated with chronic heart failure, nanodisc reconstituted APJ was used to immunize camels and generate a library yielding SDAB JN241, an effective competitive antagonist. Additionally, nanodiscs presenting the tetrameric form of influenza A's M2 protein enabled immunization of a shark species and the identification of VNAR AM2H10, which blocked M2-mediated ion influx and demonstrated antiviral activity. Meanwhile, researchers from University of Michigan demonstrated that high-density lipoprotein-mimicking nanodiscs loaded with doxorubicin can effectively enhance immune checkpoint blockade for cancer therapy. While immune checkpoint inhibitors alone benefit only a subset of patients, combining them with a nanodisc-mediated chemotherapy triggered immunogenic cell death and robust CD8 plus T cell responses in murine tumor models. Doxorubicin-loaded nanodiscs primed tumors by broadening T-cell recognition to include tumor-associated antigens and neoantigens, all without significant off-target toxicity. When combined with anti-PD-1 therapy, this strategy achieved complete tumor regression in 80 to 88% of mice with established colon carcinoma, and provided long-term protection against recurrence. This study presents a promising and generalizable approach to synergize chemotherapy and immunotherapy via nanodisc platforms. Despite their promise in membrane protein drug discovery and delivery, lipid nanodiscs face several limitations. Key challenges include the need for efficient, low-cost, large-scale production and a lack of understanding regarding their in vivo metabolism, degradation, and interactions with biomolecules, which are essential for ensuring safety and efficacy. Different nanodisc types also exhibit specific drawbacks. MSP nanodiscs cannot replicate natural membrane curvature or lipid asymmetry, affecting peripheral protein interactions. Peptidiscs are costly and less stable due to non-covalent assembly. Supposed nanodiscs are less uniform and more complex to assemble. And SMA-based nanodiscs suffer from UV absorbance, low pH and divalent ion sensitivity, reduced solubilization efficiency, and complications in affinity purification. Though polymer modifications have addressed some SMA limitations, this area remains under active development. Looking ahead, future advancements may focus on multifunctional nanodiscs with diagnostic and therapeutic capabilities, stimuli-responsive smart nanodiscs for controlled drug release, and personal nanodisc-based delivery systems tailored to individual patient needs. Moving on to computational tools for antibody-drug conjugate development. Despite decades of therapeutic innovation, cancer remains the second leading cause of death globally, accounting for approximately 10 million deaths annually. While conventional chemotherapeutics remain a mainstay of treatment, their nonspecific cytotoxicity often results in systemic toxicity, off-target effects, and the development of resistance. Antibody-drug conjugates combine the specificity of monoclonal antibodies with the potency of cytotoxic agents to enable targeted cancer cell eradication. There are 15 FDA-approved agents as of November 2024 and many more in development. Yet, the complexity of ADC architecture, including the need for precise target selection, optimized conjugation chemistry, and payload release kinetics, poses persistent challenges to rational design and efficient development. In response, researchers are looking into the integration of computational tools, from machine learning-driven antibody engineering to generate linker design and in silico pharmacokinetic modeling. Artificial intelligence and machine learning are transforming the design of development of ADCs, replacing empirical trial and error with predictive data-driven strategies. Traditional approaches have been limited by incomplete structural information, inefficient linker payload selection, and low-throughput experimental methods. In contrast, modern AI frameworks enable modeling of ADC components, predicting antibody structures, identifying optimal conjugation sites, and forecasting pharmacokinetic and toxicity profiles. Tools such as AlphaFold 3 and DeepAB support high-resolution modeling of antibody-antigen complexes, glycosylation patterns, and linker conformations. Generative AI models, including GANs, VAEs, and diffusion-based architectures, now allow the de novo design of antibody variants and linkers tailored to specific functional requirements. These advances are further integrated with high-performance computing and experimental validation pipelines, enabling rapid iteration and optimization. Building on the advances in AI-guided design, the integration of physics-based computational modeling offers complementary insight. Despite clinical success, ADC development remains constrained by a persistent lack of high-resolution structural data. This structural opacity impedes rational design, making it difficult to predict conjugation site accessibility, Recent work has highlighted the critical role of 3D molecular modeling and dynamics simulation in addressing these challenges. These enable the visualization and prediction of conformational flexibility, solvent exposure, and intermolecular interactions across ADC components. Machine learning tools, when combined with physics-based simulations, further improve the ability to identify optimal linker types, conjugation strategies, and drug-to-antibody ratios. By refining experimental hypotheses and reducing the number of necessary design iterations, these hybrid computational approaches bridge the gap between empirical assays and predictive design, supporting more efficient and rational ADC development. Expanding on the integration of structural and physics-based modeling, recent efforts have introduced deep learning architectures specifically tailored to decode the complex structure-activity relationships in ADCs. ADCNet is a unified neural network framework designed to predict the anti-cancer activity of ADCs by jointly modeling protein sequences, chemical structures, and drug-antibody ratio values. By leveraging advanced representation learning from models such as ESM2 and FGBERT, ADCNet effectively captures the molecular features of antibodies, antigens, linkers, and payloads. Meanwhile, researchers are looking into the optimization of chemical linkers. One example is Linker GPT, a transformer-based architecture trained on large-scale molecular datasets and fine-tuned for ADC-specific linker generation. Leveraging reinforcement learning, the model optimizes drug likeness and synthetic accessibility, producing structurally diverse linkers that surpass commercial analogs in key in silico metrics. However, the study remains computational. Translating these in silico gains into clinical relevance will require experimental validation of stability, biological activity, and pharmacological performance. Let's move on to the topic of antibody-antigen research. Understanding how proteins interact with each other to form functional complexes at the atomic level is key to understanding biological processes. Progress in structural characterization, modeling, and analytical sensitivity has deepened insights into binding interfaces, multivalent interactions, and epitope targeting. A recent study has revealed a surprising contributor to functional potency of bispecific antibodies, domain order. Using cryo-EM, researchers compared formats of a diabody type BSAB targeting EGFR and CD3. The X3LH format exhibited over 100-fold greater anti-tumor activity than its HL counterpart. This enhancement was attributed to a favorable inter-antigen bridging angle conferred by domain rearrangement, which mitigates steric hindrance and optimizes cellular engagement. These findings underscore the critical role of geometric alignment in designing potent bispecifics and offer a structural rationale for optimizing domain architecture in future constructs. The dynamic nature of antibody-antigen interfaces demands a detailed understanding of how structural features mediate binding specificity. A comprehensive structural analysis revealed that antibody complementarity determining regions, CDRs, especially H3 loops in VHH antibodies, exploit deep antigen concavities to achieve high specificity. These findings illuminate conserved strategies of mutual interface engagement and provide a framework for designing antibodies that target elusive or poorly accessible antigenic surfaces. Mathematical modeling also provides a critical insight into the multivalent binding behavior of antibodies and the resulting avidity effects. A recent study developed an ordinary differential equation model simulating IgG binding to membrane-bound antigens, revealing how parameters such as antigen density and antibody concentration affect occupancy and bivalent binding. The model highlighted regimes in which the avidity effect is maximized, which is an important consideration for optimizing therapeutic efficacy. This quantitative framework supports rational antibody engineering by predicting conditions that improve binding strength and functional outcomes. While traditional approaches often miss low-abundance antibodies in polyclonal responses, photo-cross-linking techniques coupled with single-particle electron microscopy have significantly improved detection sensitivity. Applied to viral glycoproteins, this method enabled visualization of weak and transient antibody interactions, uncovering previously undetectable epitopes and intermediate binding states. Such resolution is particularly valuable in early and late immune response phases and holds promise for accelerating vaccine development through comprehensive structural characterization of human and animal repertoires. Additionally, X-ray crystallography remains the gold standard for resolving antibody-antigen interfaces at atomic resolution. Through a robust pipeline involving Fab fragment production, complex purification, and crystallographic mapping, researchers can obtain precise models of interaction interfaces, including conformational epitopes. These structures inform targeted engineering, elucidate mechanisms of action, and refine epitope-based vaccine designs. The method's adaptability across diverse antigens ensures its continued relevance in both basic and translational immunology. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or on our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.